Hello, everyone. Welcome to our discussion on non allelic interactions, of which we are going to discuss the fourth one, which is the polygene or polygenic inheritance. So, in this, we are going to discuss about a condition where one character is controlled by more than two non allelic genes, and the action of the genes will be cumulative or additive. That means whenever the number of these polygenes or the interacting genes increases, its effect or the character or the trait which is being expressed also will have a quantitative effect. That means if you're having one, it will show a particular, for example, if it is the case with color, if you have a one if you have one of that particular gene, you have a light pink color. If it is two such genes, you have a dark pink color. If you're having three genes, you have dark, and it goes on. So that is what is called as a cumulative effect. That means it depends upon the trait, or the phenotype of the trait will depend upon the number of genes present in that particular individual, and that is what is called as a cumulative effect. So here we are going to discuss about many genes controlling a particular character. You will have multiple genes, but all of them will be controlling a particular character. As a result, the phenotype will depend upon how many genes are present to control that particular character. So such genes are called as polygenes or multiple genes, and their mode of inheritance is called as polygenic or multi-factor inheritance. So there are certain traits which are exhibited in the quantitative variation or a quantitative fashion, or in a polygenic manner. They are height. So that is true because we are having two extremes of height. You have a short stature, which is of four something, and we are talking about the normal heights, not with some syndrome or some malformations. In the normal height individuals itself, normal height means you cannot say that all normal individuals are having 5 feet. It is a range. It ranges from either 5 feet or 4 feet to 7 feet or 6 feet. So in that 6 feet, you will have from if you start from four, you have 4.1, 4.12, 4.13. So it is a range. If you take a population, you get a range of that particular phenotype. The same is with skin color. The skin color is also a range from dark to light. We have different shades, which is the intermediate of this particular shade. So all these are examples, height, weight, etc. All these are examples of the so here you have a single trait, which is skin color or height or weight, and that is going to be influenced by a number of genes. That is why it's called as multifactorial or multiple factor inheritance. So such traits you are usually found to be quantitative rather than qualitative. Quantitative in the sense the depth of expression of the particular trait will depend upon the number of genes that particular individual carries. And polygenic inheritance are usually referred to as quantitative inheritances also. So these continuously varying traits are due to the joint action of several genes, that is called to be additive or cumulative. It differs from classical Mendelian pattern in that the entire range of variation is covered in a graded sequence rather from one parent to the other. So in Mendelian inheritance, uh, we talked about distinct characters. That means you have a white flower plant, you have a red color plant, then you get either red, or if you're having allelic interactions, you get pink, etc. So you clearly define that particular what you get. But in such cases, you cannot define that exact thing. It should be in a particular range. You have a certain chart to compare, and from that chart, you have to take it in. There will not be distinct, clear-cut traits when you come to polygenes. It will be a varying range of traits that can be expected for an offspring. There is no clear cut white or black. You get shades of white and black in the shades which is called as gray. So that is what is called as polygenic or polygenic inheritances. So that is the one which you have a difference from classical Mendelian inheritance. So many scientists also believes that Mendelian inheritance also shows also follows polygenic inheritance, in the sense that when Mendel studied the height of plants, every plant didn't have the same height. So what Mendel did was he put a cross mark or a border line. So if the particular plant is above or up to the borderline or above, he considered it as tall. If it was below a particular borderline, he considered that as short. So every plant was not short with the same height, and every plant was not tall with the same height. So that's why many scientists also argue that such traits which Mendel clearly defined as tall and short might not be exactly tall and short. So all these can be categorized under this particular type of interactions, which is called as polygenic interaction. So here we take the skin color in man. So skin color in man is mainly influenced by the particular pigment which is called as melanin, which is deposited under the skin. So black color, skin color is due to the presence of melanin, and white is due to the absence of it. So the total number or the amount of melanin pigment depends upon the number of genes. So if you have the maximum number of genes, you will have the darkest of the skin color, which is the utmost dark, which can be called as a Negro race. And if you are having the alleles which do not contribute to melanin, that means if you are having alleles which do not produce melanin, every allele is in the sense that they have not have the capacity to produce melanin. You will have a skin color which is called as white, which is called as European or the Caucasian type of race. So the Negro race have a heavy deposition of melanin, while the European race have the least amount. And marriages between a Negro and white race will result in a children. So that is shown here. This one is white. You can see here. All these they do not have the capacity to produce melanin. As a result, they are very white. All these are dominant genes. Hence, all these, because they are multiple genes. So this is just an example. Here we have taken three genes, and actually it is said that for human skin color there is something from 4 to 7 genes which are responsible for producing human skin color. So for the sake of our discussion, we have taken only three, because if you take more, the Punnett square, the columns and rows in the Punnett square will go on increasing. So for our discussion, we have limited this to three, and don't consider it as these three genes are responsible for producing the skin color. So it might vary. It is always greater than three. We have considered this only for the sake of discussion as three genes which is responsible for human skin color. So in this case, this produces a heterozygote. Here you can see. So in this heterozygote, the heterozygote, if you compare the heterozygote and this dark one, you can see that it is having half the number of genes. Half the number of genes that produces melanin. Here all the genes can produce melanin. Here no genes can produce melanin. When you come to this heterozygote or the intermediate one, you can see that it is having half that of this particular one produces melanin. As a result, it is having this it is not dark as this. It is having a lighter shade than this, and this particular shade in human skin color is referred to as something which is called as mulatto. Okay, and this mulatto, if you cross two mulattos, you have an F2 generation. In F2 generation, because we have three genes, you will have such number of or combinations of sperms and eggs can be produced from this. As a result, you will have 64 different shades of skin color, and you can see the shades depends upon the number of dominant genes present. If you take from here there is zero, which is white. If you have one dominant gene, it will produce some amount of melanin. If it is two, it is producing more. If it is three, it is same as that of our mulatto. If it is four, you can see the difference. between 3 and 4, it is slightly darker. If it is 5, it is darker than 4. If it is 6, all genes are dominant, it will be having the darkest of the skin color. The complete, you can see this is what is called as a quantitative trait. So it is having, so this 0 is one parent and the 6 is one parent. So 0 is white, this is dark, and we have the offsprings in all the different shades. So this is same with the height, intelligence, then whatever the weight, etc. So all these follow this type of inheritance, which is called as polygenic inheritance. So with this, we come to the end of polygenic inheritance and the example of skin color. And the next is transgressive variation, which is something which is related to polygenes itself. So in polygenic inheritance, the or quantitative inheritance, sometimes the offsprings do not show a continuous variation, as discussed in polygenes. Some individuals may exhibit more extremes than either of the parents or grandparents. That means when we explained polygenes, we said that they have a gradation and that will be limited by the shades of the parents or grandparents. That means if you take this column, the previous one, you can see that this is the limit that we have set, and this limit is that of the parent or the parent or that of the grandparents. But in some cases, or in most of the cases of polygenic traits, it does not limit with that of the parents. You can see the phenotypes which is more lighter than white, or if you take the skin color, more lighter than the white of one of the parent, and you can see the darkest of the color or darkest of the skin color than that of the negro or the dark one. So that means extremes of either of the parents or grandparents can be seen in some cases, and such extremes are produced, that means the extremes other than that of the parents. If you are having a phenotype which are extremes of the parents, that means usually in polygenes we expect that the range will be between the parents in the sense between white and negro. So that is the extremes we have, and that is the parent we took. So we expect this to be between the range to be between these two particular phenotype, but it is said that it will not be in that range. In some cases it can go beyond, it can go below white as well as it can go beyond the darker. So dark can be darker and light can be more lighter. That is what is called as extremes. And such, whenever you have or you come across such extremes in quantitative traits, such extreme variations are called as transgressive variation. So it can be seen in eye color, skin color, intelligence, etc. So that is what is called as transgressive. So it is a variation from the normal parental phenotypes or parental ranges are called as transgressive variations. Coming to the last topic of our discussion on non-allelic interaction and also gene interaction, this is pleiotropism. So this is something which is, you can call it as opposite to polygenes. Polygenes, we said that we have a single trait controlled by many different genes. Here it is opposite. You're having a single gene. It is controlling many traits. A single gene affecting many traits is called as pleiotropism. A single gene controlling or influencing two or more unrelated characters is called as pleiotropism. We'll take the examples and discuss. So in Drosophila, there is a recessive gene for vestigial wings. So vestigial wings occur in, or vestigial wings is seen in Drosophila when they're having this homozygous recessive gene for vestigial wings. And whenever they're having vestigial wings, they also will be affected by many other factors, or many other structures will also be affected with this particular gene. That means eye color will be affected, the position of the balancers, position of the bristles, the structure of reproductive organ, fertility, lifespan, etc. That means when one gene was affected or one gene became recessive, it is affecting not only a single character but many other unrelated characters are affected. And hence you can consider that that particular gene is affecting all that unrelated traits, and that is what is called as pleotropism. The same can be seen in man. The example is of the disease which is called as phenylketonuria. As discussed in the earlier session, phenylketonuria is a metabolic disorder where the amino acid, the phenylalanine, is not being metabolized. So, what you expect is that metabolism of phenylalanine does not occur. As a result, phenylalanine can be seen in blood, urine, etc. In everything, you can see this phenylalanine. And apart from that, it is also seen that phenylalanine can be seen in the cerebrospinal fluid, blood, etc. Apart from this, individuals have short stature. So, a gene which is affecting metabolism is affecting height. Another thing, mental retardation. So, it is also affecting neuron development. That particular gene which is affecting metabolism causes pigmented patches in the skin and non-pigmented hairs and eyes. So, and this particular gene which is causing a particular damage in metabolism, the gene for phenylketonuria, that is causing many or affecting many unrelated traits, which also point to the fact that a single gene is controlling many other unrelated traits or characters. So, with this, we come to the end of this particular module, which is gene interactions. In gene interactions, we discussed about allelic interactions, the three major or the four major types of allelic interactions. We discussed about the dominant different dominant interactions, followed by codominance, then incomplete dominance, and lethal genes. After that, we discussed about non-allelic interactions. Complementary genes, supplementary genes, epistasis, and the last one, polygenes and pleiotropism. So, with this, we come to the end of this particular module. Thanks for hearing.